Alrighty. Let's start from where we were last time. Apparently, Oscar's already exhausted. I'm current. All right, so here is where we were last time in our discussion about mobilization. Let me try to even this out. So we were talking about ways in which organisms can try to take advantage of metals that are not necessarily in their bioavailable form. So what do I mean by bioavailable form? So in forms that can be digested, metabolized, and potentially transported into cells. And so there are different ways that organisms have adapted to be able to extract what they need. We talked about the idea of chelation being a method of doing so, how chelation, and especially with a ligand that has a strong affinity for a metal, can enable release of a metal. But the very important thing is that there needs to be a moderate amount of lability, the ability to be able to release that metal if it's actually necessary for use. Then you have the two other features, redox, where you can fine tune the oxidation state, either increase or decrease it, and that allows for solubilization, but also recognition by certain transporters. And what we just looked at in the example, acidification, where we looked at an organism solubilizing nutrients, not necessarily metals, but in this context, if you're trying to acidify metal, typically the acidic environment will protect the metal from hydrolysis. Hydrolysis is known to be able to keep metals in their insoluble form, so the acidification is a way to get the metals as accessible as possible. All right, so we talked about a few different coordination modalities that are present in molecules called siderophores. So these are molecules that are predominantly used by bacteria, but there's also evidence for it in fungi and some plants, marine organisms. So the idea is that they have these coordination modalities, these moieties. And here are the example of catechol. Here's the example of hydroxamic acid. And if you look closely at these molecules, you have the capacity of binding at multiple sites, and therefore these particular molecules serve the capacity as chelators. And so here, as I was showing you previously, are the crystal structures that are associated with the coordination of these particular molecules to iron. That's the primary metal that these particular molecules are used in order to mobilize them and to be able to extract the metal and use them. And so a bit of information is that there is a sense of stereochemical preference. So how these ligands wrap themselves around the metal can afford some amount of selectivity in terms of the uptake of these particular metals or metal compounds. So at the very end, we discussed this idea of the binding constant. So we were looking at what is referred to as a beta value. A beta value is simply a K value, but it's defined for something that represents multiple steps. And so when we're talking about these type of ligands, as we already saw, they have the capability of binding at more than one site. As an example, the enterobactin siderophore, when it coordinates to iron, it actually saturates the coordination sites for iron, and iron 3 tends to prefer coordination number 6. And as a consequence, what you have is a coordination modality where 3 of the catechol moieties will coordinate to the metal center. So that gives you your very favorable coordination number of 6. The very last thing that I wanted to point out, and just to reiterate what we discussed previously. So these values that we see here are a comparison of how strongly those ligands bind to iron when they form the complete coordination complex. And as you can see, enterobactin has the highest affinity in this list of siderophores. But then another value that I pointed your attention to at the very end was this value of PM. And so what we're going to do, we're going to see how that value is comparable to the parameter pH, where pH is a measure of the amount of free protons in solution, right? So we have the sense of how pH can relate to the acidity of a solution, whether it's neutral or whether it's basic. Well, in this concept, what we're measuring is the amount of metal that is not ligand bound. So as you can assume, a ligand that has a very high affinity for that particular metal will result in a lower amount of that metal accessible to solution. All right, so here is just basically the measure of that free metal in solution, and that's defined as metal in aqueous conditions. And so the higher that PM value is, the lower the concentration of free metal in solution. So that gives us a parameter that we can use to get a general sense of the available iron in solution that is not complex to the siderophore. All right, so let's talk a little bit about the how the iron siderophore is actually transported. So just an example, as we're looking at the transport as it pertains to a type of bacteria. So it turns out that bacteria have evolved very high affinity outer membrane receptors. And so these receptors serve as what we have already been referring to as transporters. Their focus is to bind the iron siderophores and transport them. And so what we're looking at here is a representation of a carrier. And so this is just a cartoon depiction of a type of bacteria. So we're not actually focusing on a specific bacteria, we're focusing on the representation of a gram-negative bacteria. And so what we see here is just a composition where we have an outer membrane and the corresponding constituents of the outer membrane. So we have a certain number of transporters that are available. And then we have what is called the periplasmic space. And then you have what is the inner membrane before you even reach the cytosol of the bacteria. So I'm just going to briefly talk about how a particular siderophore is transported into across the outer membrane, the periplasmic space, and also the inner membrane. All right, so in terms of just the properties, we're looking at a substrate-specific carrier that's going to engage in an energy-dependent active transport. So remember this terminology in terms of the energy required in order to transport a particular solute. In this case, we're referring to this compound, which is ferrochrome. So this is a type of siderophore. And if you pay attention to the coordination moieties, right? So these are the hydroxymate moieties that are coordinated to the iron, satisfying the coordination number there. All right, so let's look at what happens in terms of the transport. So there exists a transporter that is a receptor that would recognize this particular siderophore when it's bound to the iron. So that's an important event. The configuration of the compound is what's recognized by this particular transporter. And so when we're looking at this FHUA transporter, in terms of its structure, what you can see here is a pretty complex structure. It consists of a 22-strand membrane spanning. As we know, this is a transmembrane protein, and it's antiparallel beta barrel in terms of its actual structure. And so what is governing the movement of the actual compound is a conformational change that the protein must undergo. So in the apo form, when I'm saying something that's apo, that means that it's metal free. It doesn't actually have the metal bound. It is apo form. You have a conformation of the protein. It has a structure that basically blocks anything from coming in. But then when you have the contact between the complex, in this case, the ferrochrome, with the receptor, that induces a conformational change. That conformational change will then open up the protein to be able to transport it through. All right, so let's see what are the subsequent steps of that process. So we're here so far. We have our ferrochrome iron. It gets recognized by the FHUA transporter, and that's at the outer membrane. Okay, so then what happens next? So what occurs then after, as a consequence of that conformational change, the compound will go through, okay? And so what eventually happens is released into the periplasmic space, and there is actually recognized by something else that would actually help to facilitate the further transport through the inner membrane and into the cytosol. So the transport of the iron across the cytoplasmic membrane can be in the ionic ferric or ferrous form. Okay, so what do I actually mean by this? So what I mean by that is that at this point, your iron can be liberated from the siderophore and potentially be transported. So it can be transported as either the iron 2 form or the iron 3 form. Okay. Another likely route is as a complex, the iron 3 siderophore complex. Remember, that's a high affinity complex. It gets recognized by an ATP binding cassette, or referred to as ABC transporter system. And that system will then enable the further transport of the complex through the inner membrane. All right, so I wanted to emphasize that just to give us a perspective of this idea of how bacteria have evolved a lot of receptors to be able to recognize this big family of siderophores. So that's just one example, but there are many different siderophores that have been developed. And what's the purpose of this? Why is this redundant? So the redundancy is important because of the competitive nature of bacteria to be able to hold on to the nutrients, in this particular context, the iron. And so it has evolved many receptors and also the small molecule siderophores to be able to exploit the iron that's available in the surroundings and compete with other microbes. So it turns out that other organisms take advantage of these systems. All right, so here's just in a representation of a yeast that can take advantage of the siderophores to be able to also exploit the iron that's bound to the siderophores. So let's look at this example here. So it turns out that certain receptors in organisms that actually do not express, or they do not produce, not express necessarily, do not produce their own siderophores. They don't have the biochemical machinery to be able to synthesize these compounds. However, they have evolved the mechanisms to be able to recognize them. So here's just a representation of this particular yeast, and I referred to this yeast previously when we were looking at it as a zinc transporter. So what this particular yeast does, it takes advantage and it preys on the bacteria and exploits the iron siderophores that are actually produced by the bacteria releasing its siderophores. And as a consequence of that, it can actually uptake those siderophores. And you can see sort of this complex system that it has evolved to be able to just take advantage of those molecules, something that it itself does not produce. All right, so this is just a representation of how other organisms can also mobilize by being able to recognize molecules that are not innate to them, but they have receptors that can actually recognize them. All right, any questions about this? Any doubts? Brenda, anything? Very good, all right. All right, so let's continue on our discussions. So I'm gonna just switch for a moment. Actually, let me switch here. All right, so we're going to continue this exploration of transport, but we're going to tailor this to the transport of iron, and we're going to look at this in the perspective of how mammals in this context, we're mostly focused on humans being able to transport the iron. So I'm actually going to skip over this slide here. All right, so I'm going to emphasize iron in terms of its value in living organisms. So here we see a representation of iron that is represented by the proteins that are either transporters of the metal or depend on the metal to be able to facilitate certain key enzymatic activity. And so here's just the proteins on the left and the corresponding functions. So we're all aware, and I've emphasized this a few times, of the importance of hemoglobin and even of myoglobin, as these are heme-containing proteins that take advantage of the iron that's present in that actual small molecule center to be able to facilitate oxygen transport and oxygen storage, right? So it's in the iron 2 form that these proteins are able to have a high affinity for the iron. But we also see other types of heme-containing proteins like the cytochromes that have this capacity to facilitate enzymatic activity, even though they're using the same type of heme moiety. What surrounds that heme moiety enables certain types of activity. So here, for instance, the cytochromes have an important role in terms of electron transport, ATP synthesis. And if you probe more deeply, cytochromes actually play a very critical part in terms of the survival or death of our cells, as the release of these proteins, or cytochrome C, is critical to the induction of a cell death process known as apoptosis. So cytochromes play a very significant role that goes just beyond just the function of the metal itself. All right, so I'm emphasizing this just so that you get a flavor of where you can find iron within proteins, what types of functions that it enables. And I've emphasized so far these proteins that are heme containing, but there are plenty of proteins that do not have that type of moiety that have evolved this capacity to bind iron, whether it is for transport purposes or whether it's for storage purposes. And what we're going to focus on today is this idea of how do we go about mobilizing but also transporting iron. All right, so this is something that I do in my undergraduate inorganic chemistry course. I do this experiment. I'm not sure if you've done this before. So what I do is I take a box of cereal that is fortified with iron. And so it turns out that these cereals are fortified by little filings of iron. They're neutral form. And in this case, we're taking advantage of the magnetic property of the metal. And so what I do is I give everyone a bag of cereal, we put some water in it, and we mush it up to homogenize the sample. Very similar to what I discussed previously, this idea of trying to extract. And then what happens is by using the magnetic barb, you can have this attraction to the magnetic iron filings in there, and you can grab a lot of iron. You can't see it there, but you get a significant amount of iron. And so it's quite nice. Total cereal is one of those that have a very high abundance of it. So anytime you see a cereal that says 60% or more of the daily serving of iron, that's going to have a lot of iron. And you can actually do this type of experiment very nicely. All right, so clearly food is a source of iron. But you think about, in this example, you see the iron is in a filing form, it's a solid form. So sometimes when students see that, they get a little scared. It's like, is that what I'm eating? Well, yes, that's what you're eating because your body has the capacity to metabolize it. Otherwise, it's not going to be something that's essential for in terms of your delivery of your amount of iron that you need. So what I want to talk a little bit about is this idea of how do we try to extract the iron that's in our food to be able to take advantage of it and transport it ultimately into our cells to be able to use it. All right, so this is essentially this idea of ingesting iron and the bioconversion of that iron into a form that we can use. So the elemental iron, maybe not necessarily in that form, something that's soluble or useful, but our body has the capacity of handling it. So this is the idea of ingesting iron, either in the iron filing forms or even as in proteins as heme, right? So that's the iron 2 form, or even can be an iron 3 form, or even non-heme iron 3. All right, so the thing that we need to keep in mind is that in the process of digesting, is this what occurs in our digestive system. What we have is an increase in the acidification. So we see a decrease in the pH. So an increase in the acidification, that results in the mobilization that we need. Right? So that's the themes that we already have touched upon, but we're looking at it in the context of us. All right, so the very critical thing is to be able to convert that iron into a form we can use. And one of the major ways is by converting it into the iron 2 form, so that ferrous form. And so let's look at some of the ways that we can do this. All right, so in terms of the iron that's supplemented into cereal, so that would be elemental iron. So for us to be able to take advantage of that iron, there has to be a sort of redox process. So we have the iron in its neutral form. We have the acidification that's enabling some amount of the mobilization of this metal. But as you can see, there is a change in the oxidation state. All right, Brenda, what is the change in the oxidation state in the iron? 2+. So that means that we have a loss of what? 2 electrons, right? So what's facilitating that is the actual protons. If you look at the composition of the protons, it goes from being H+ to then being in this neutral form in hydrogen gas. So you have the hydrogen that's facilitating this change in oxidation state, okay? And you also see that there's a release of hydrogen gas, right? Another thing, let me actually clean this up a little bit, is that we can extract dietary iron 3, okay? So we extract the iron 3 that's available, but then that iron 3 has to be reduced to its iron 2 form, okay? The reason why, and the emphasis on iron 2 is because of its relative solubility. And so how much we can get out of the nutrients where we pick up is how much we can potentially be able to deliver into our blood. Obviously, there's going to be a cap about that, and we'll talk about that in a moment. All right, so you have iron 3. And so now we're looking at iron 3, which is now 1 electron even less, so 3+, getting reduced to the iron 2 form. And so here you have a number of reducing agents like a ferrireductase, which is a protein, okay? It's something that's a cell surface protein that can facilitate the reduction, or you have small molecules, such as ascorbic acid. I know that Oscar has worked with ascorbic acid for this particular purpose of it serving as a reducing agent. All right, so in its iron 2 form then, now you have the iron that's accessible to be able to be taken to the intestine, to the intestinal cells. And so a protein that can participate in this process is this protein called gastroferrin that can bind the iron and then deliver it to intestinal cells for it to be uptaken. Another possibility is that maybe you don't have free iron available, right? Because free iron, this is something that's possible in the acidic environment in which it's placed. But perhaps you have it in the heme form that's extracted from, let's say, if you are meat eating, that's extracted from the meat that you've eaten, right? And so it turns out that there are cells that have, that there are receptors on cells that can recognize heme and can facilitate a receptor-mediated endocytosis of the heme group. All right, so once you have the iron available, let's say iron 2 available, we're not talking about the heme form, there's actually a protein that will now recognize that iron 2 and facilitate its movement. And so this protein that I'm referring to is the divalent metal transporter. We're going to see this protein mentioned again a little bit later on as well as we look at a little bit further in terms of the transport of the iron. Well, here we're looking at the movement of the iron from the intestinal lumen into the enterocytes. So these are the actual intestinal cells. And so we're going to see how this transporter, the divalent metal transporter, facilitates this movement. So let's talk about some of the features of the DMT1. It is a secondary active transport symporter. So basically here, we're seeing that there's a movement of iron that's coupled with a movement of protons. So that's going to be an important feature. So it uses a proton concentration gradient and the electrical potential of the membrane to drive iron to uptake into the actual cellular environment. So this is what's facilitating that movement into the cells. All right, so let's look at some of these features that we talked about. So I already mentioned this idea of heme being recognized by a receptor. So here's just a cartoon representation of an enterocyte. Okay, this is an intestinal cell. We also have iron getting reduced to iron 3. So the example here is getting reduced by some sort of protein that's located at the cell surface. So this ferri reductase, okay? And then once it is an iron 2 form, you have the divalent metal transporter that's facilitating the secondary active transport of the iron. All right, so then what happens next? Now we're inside the enterocyte, we're here, okay? Whether it's going through this route or whether it's going through the heme route, okay, now we have the iron 2. So you see that there's a protein here that's called ferritin. We're going to talk about this in a little bit in the slides ahead. As proteins responsible for the storage of iron. So I'm going to ask you, at the very beginning or the second lecture, I talked about a storage protein for copper and zinc. Do you remember what protein that was? Anyone that's online, does anyone remember the protein that said there's a storage for copper and zinc? All right, so the protein is metallothionein. So those are the proteins that have a very high cysteine count. Because of the cysteine count is very favorable for binding of the zinc and the copper 2, which are more intermediate metals. So we're going to talk about ferritin again in a little bit. All right, so what happens next? So you get iron that's going to get transported through this porin, this transporter that's going to release the iron. So that's coupled to an event where the iron that was in its iron 2 form, again, very soluble, now getting oxidized to its iron 3 form. So that's something that's not very intuitive. Why would that be a requirement? All right, so let's wrap up a few things that we just discussed. So we have the iron 2 that's going to get exported through the membrane and is going to go through this ferroportin. That's basically a transporter that's going to release the metal. Then you have this really important enzyme that's called hephaestin. This enzyme is copper dependent, is a ferroxidase. So as the name implies, it oxidizes the iron and it transforms it into its iron 3 form. There are actually a number of copper-dependent ferroxidases, and there's a family of these proteins. And so here you can see there's this correlation between copper and iron in terms of the transport of the two, and also their functions. They're correlated. All right, so you have this other protein, ceruloplasmin, which is another ferroxidase, and is a major copper transport protein. All right, so that's basically what we see here. But what we're going to focus on now is this process here, a protein that is called transferrin and how it plays a role of binding that metal and facilitating its further transport through the body, the distribution. All right, so let's look at this for a moment, this idea of the solubility of the iron. So as I already mentioned to you, iron 2 is quite soluble, whereas iron 3 is not. And so here I'm just showing you this idea of the iron getting oxidized, and then at a pH of 7.4, and I'm highlighting that pH because that is the pH of serum, the pH of blood, we see that there's going to be a sort of precipitation event. So here's just a representation of this, where you have iron interacting with hydroxide that's available at a pH of 7.4, and that's leading to this event where you form iron hydroxide as a solid, so it precipitates. So one of the key things is that this introduction of the iron into the environment of the blood would result in a precipitation. Okay. So the question is, well, how much soluble iron would there be? Why is there a requirement for a protein to bind that metal? Well, if we were to try to calculate the solubility of the iron and taking this species, and I'm just giving you one example of a species that can form. You take that and you look up and remember this from general chemistry, the solubility product that gives you the relative solubility of a given species, right? You present this in terms of the context of the dissolving with ionization. So you have basically the ions separating, so you have the iron being released. You use this information to determine how much soluble iron there really is. So here, I looked up this number, KSP of 10 to negative 38 molar. So with this information, and also take into account the pH, remember in this expression, we have the concentration of hydroxide. If you have pH, you can correlate that to hydroxide concentration. You can calculate how much iron would be soluble. So I did this for pH 7. I could have done this for pH 7.4. It doesn't really matter so much. The take-home message is the following, that you'll get a concentration of iron that's 10 to negative 17 molar. So that's a concentration that's extremely low compared to what you actually see of iron in our blood or even iron in our cells. So if you're looking at iron in just the fluid part of blood, we're talking about micromolar. That's 10 to negative 6. If you're talking about iron that's in the cellular or the cytoplasm, that's still iron at 10 to the minus 6. So that concentration of iron is much lower than what we actually have in our body. All right, but question is then, why oxidize that iron 2, especially for something that is, from all perspectives, something that would be less bioavailable? Well, it turns out that iron 2 is something that, just as it's very soluble, it is also prone to a lot of reactivity. And so our body has a mechanism for controlling that reactivity. Iron 2 is well known for generating what is known as reactive oxygen species and also can generate reactive nitrogen species. For the reactive oxygen species, or commonly referred to as ROS, it can do so via Fenton-type chemistry. All right, so if you have this freely available in our blood, the problem you're going to have is you're going to have the uncontrolled production of this ROS formation. And so with enough of this formation, and that's not to say that ROS is not important, but having an excess amount of it can be something that will induce irreversible damage, and it can lead to, in the context of our cells, oxidative stress and cell death. So it's something that becomes quite toxic quite quickly. So in that context, then, the transformation of iron 2 to iron 3 is sort of a regulation step. It basically controls that metal for being able to produce those type of species. But clearly, that iron cannot be released and left like that, because if it was left like that, you would have a precipitation of the metal, and now that metal is not actually available for use. So this is the perspective of iron released into pH 7, aqueous solution, precipitates. If it were in this iron 2 form, we have this reactive oxygen species. But the reality is not that. The reality is that you have a protein referred to as serum transferrin. Commonly it's referred to as transferrin. I'm saying serum transferrin because of where it's located, which would be in the blood. And it has the capacity of binding 2 equivalents of the iron in the iron 3 form. There's also evidence to suggest that there's a transient amount of small molecules that can bind the iron, and when they bind that iron, they help to deliver it to the protein. But this route of transport is going to be something very quick. Okay, so it needs to be something super fast so that the ligand, whatever it is that binds that metal, has an affinity, but also has a relative ability to release that metal to be able to bind the transferrin. So it turns out that the iron that is released into our blood will be virtually 100% serum transferrin bound. Okay, and that would be the iron that's in our fluid blood. That's not talking about the iron that's in our red blood cells, right? That's iron that's trapped in the hemoglobin form. But what we're talking about was actually fluid, and that's going to be in the transferrin form. Sorry, my slides get stuck sometimes. All right, so let's focus on this protein, and here's just a representation of the crystal structure of this protein. And what we're looking at is the nature of it. So it is actually a bilobal protein. Okay, so let me just move that out. All right, so what do I mean by that? So you see that it forms this type of structure that is bilobal, okay? And it actually is part of a big family of proteins called the transferrins, okay? So it turns out that, or it's believed, originally the transferrins were monolobal. So they only consisted of one protein lobe that was about 40 kilodalton, and that it evolved to be the bilobal form, to be able to, in theory, maximize the amount of iron binding. Today, there are some representatives of the monolobal protein still existing in some of the simpler organisms, but in our body, in the mammals, you're going to find the bilobal form. Okay. All right, so what are the functions that serum transferrin plays, but in general, transferrins can play? So they can participate in the process of iron transport and in facilitating homeostasis. So this is a term that I introduced in our very first lecture, this idea of balancing the optimal amount of iron. They also can participate in bacterial stasis, and something, a theme that we're going to return to later in the course, this idea of the transferrin and other molecules helping to protect us from bacteria, pathogenic bacteria that want to exploit the iron that's in our body. So what happens is that you have transferrin and even other members of the family, lactoferrin. So lactoferrin is present in milk, right? So these type of proteins will grab onto that iron and not make it accessible. So in that way, it protects us from bacteria being able to pull the iron away. So in terms of the affinity, so here is the affinity constants of iron bound to the two lobes. All right, so I want to ask us, just to remind us, when I refer to the C or the N, what am I actually referring to in a protein? Do you remember? The terminals. So in this context then, when I say C lobe and N lobe, I'm referring to the lobe that is towards the C terminus and the lobe that's towards the N terminus. And so what we can see is that this affinity constant, they are similar but not identical. And in fact, this is log number, so log K. So they're different by an order of magnitude, so 10 times, right? So it's interesting because when we look at the metal binding site, the primary coordination sphere, coordination sphere, they are identical. So what do they consist of? Well, they consist of two tyrosines, they consist of a histidine and a aspartate, and there's a small molecule carbonate, which its function is to help satisfy the coordination. So if you look closely, this iron that's here has a coordination number of 6. So the function of that anion is referred to as a synergistic anion because it helps to stabilize the metal. All right, so let's look at some of the features, and this is going to bring us back to some of the fundamentals that we have already discussed. So some of the features of iron binding to this protein, how can we tell that the iron is actually there? So one simple way is by using UV-vis spectroscopy. And we talked about features that correspond to the electronic transition. So here, when we look at the protein, the apo form, apo means metal-free. Let me just write that there, metal-free. And hollow means metal-bound. All right, so in the apo form, we don't see any characteristic absorbance between 400 and higher. So this is completely flat. However, when the iron is bound to that protein, we actually see the formation of this absorbance here. So what is this absorbance due to? It turns out it's due to a ligand to metal charge transfer, or LMCT, that occurs at 470 nanometers. And it's a consequence of the tyrosines that are bound to the iron. And it produces a very characteristic pink color. And so we can measure the extinction coefficient, because remember, extinction coefficient tells us what type of transition we're looking at. So you can look at this from the perspective of the protein, right? So we need 2 irons per protein, right? So if you look at it from the perspective of a protein, right, you're going to have just one equivalent. So the extinction coefficient will be 5,000 per molar per centimeter, which is characteristic, right, greater than 1,000 for a charge transfer. But if you look at it from the perspective of the iron, you have two equivalents of the iron. This number is going to be half, right? So that'd be 2,500 per molar per centimeter. Either perspective is still something that, from our understanding, is something characteristic of a charge transfer. All right, so let's define some of the features of that metal affinity of the protein. So just focusing on the protein as a metal binder, what are the things that we can say about it? So we know that it can bind iron. It can bind iron at a high capacity. And iron, from our perspective, when we discuss some of the fundamental coordination chemistry, is a hard metal. So this was done in 1996. So you can see how long ago this was done. A study was to establish a correlation between the affinity of the protein for different metals. So you have different metals that are represented here, and their general affinity for hydroxide. But why hydroxide? Well, remember when I was discussing the affinity, as you can define it for Lewis acid and bases, it's defined in terms of a relative scale. So it turns out that using hydroxide is a representation of the measure of something that can be considered a hard base. Okay, so it turns out that there is a linear correlation between the affinity of hydroxide for metals with the affinity of the transferrin for metals. And here we're just looking at the log K1, which means the primary binding site, not the second site. And you see the linear correlation. And what that indicates, as we can already speculate, that the serum transferrin metal binding site is a hard metal binder, or you can think about it in terms of its Lewis property is a hard Lewis base. All right, so additional features. So let's compare the structure of apo and hollow. And what we can see, here we have the C lobe, and here we have the N lobe. Well, we have this very profound change in the conformation of the protein when it becomes metal bound. So you see the metal here, and you see that there's a significant closure of the lobes. So there's this 49.5% closure of the C lobe, and 59.4% closure of the N lobe. So it's commonly referred to as sort of this Pac-Man-like feature when it grabs onto the iron. So what is the long-term effects of this? We're going to look at that closely. All right, so we see this change that's on a tertiary level. The protein becomes more compact as it grabs onto the iron. So it turns out that when iron binds, it actually stabilizes the protein. So here's the effect that the metal has on the protein. We already discussed what the protein does to the metal. It increases solubility, it improves its relative bioavailability, protects it from hydrolysis, but the iron also serves a function for the protein as it actually allows it to be much more stable. All right, so why is this happening? So here we have a representation of the N lobe, and what we're focusing on are the residues that correspond to the metal binding. And let's see where they're located. So over here, what we have are the two tyrosines. They're located at the site of the N lobe that's referred to as the N2 subdomain, okay? And then we have the aspartate and the histidine, although it's not as easy to see here. These are on the N1 subdomain of that lobe. So you can imagine if you put the iron right in here, what's going to happen is you're going to have this grabbing of the amino acids onto the metal, plus the carbonate coming in, although that's not depicted here, helping to stabilize this. And so you have something very similar in the C site. You have the one site, the C2 subdomain that has the tyrosines, and then you have the C1 subdomain that has the aspartate and the histidine, a very similar phenomenon there. All right, but let's go even further than that. So here we're looking at now more extensively beyond the primary coordination sphere, because the stability that's afforded by the binding of the iron, but also very importantly, why there might be a difference in terms of the affinity for the iron has to do with the secondary sphere coordination. So what do I mean by secondary sphere? I mean that anything that's surrounding the primary coordination sphere, whether it's interacting with it via some sort of non-covalent interaction. So on one level, we can see where we have an interaction that may be this modulating the stability of this. And that has to do with the carbonate, which is the synergistic anion. It turns out that the carbonate has its own little binding pocket. It has an affiliation with the arginines that are located nearby the metal binding site. All right, so the arginines at the typical pH that we're talking about here are protonated, right? And so they're going to have this electrostatic attraction to the anionic carbonate, right? So you have stabilization that's afforded by the carbonate, coordinated directly to the iron, but also interacting with his own little pocket. We also have additional features in the secondary sphere that's enabling stabilization. And so we're going to talk about these features and what they do. All right, so let's look at the end lobe, and we're focusing on what is referred to as the dilysine trigger. So lysine is something that we know based on its pKa is going to be protonated, especially in the context of pH 7.4. So just want to remind us about the pKa of lysine, also remind us about the pH of blood. This is where the context of the iron binding. Another thing that I want to remind us, and let's clear this up so you guys can see this. Think back to biochemistry when you were talking about whether a residue is going to be protonated or not. It depended on the pH relative to the pKa. So if your pH is below the pKa, that will indicate that your residue is going to be protonated. And it will be fully protonated if the pH is 1 unit lower than the pKa. Right? So that's just a reminder. But then the opposite. If the pH is higher than the pKa, your residue is going to be deprotonated. And if it's 1 unit higher, it's going to be fully deprotonated. Right? So that's something that's important to recognize. So at a pH of 7.4, we would expect lysines to be protonated. But it turns out that when the iron is bound to this metal binding site, these two lysines, so here at the end site, lysine 296 and lysine 206, well, one of them is actually deprotonated. And so that's actually a really important feature that metals have. They have the capacity of deprotonating things. And so because of that, the loss of one proton, what we see is this interaction between the two lysines via a hydrogen bond. So you have this hydrogen bond network that's facilitating a stabilization event. Okay? So that's a product of the iron binding that's facilitating a loss of a proton, but the loss of that proton enables those two lysines to basically close up the site even more. And it turns out that this hydrogen bond is even stable at a pH of 5.5. And so I will, in a moment, talk about why pH 5.5 is important, because we're going to talk about the process of endocytosis and where this pH change is actually relevant. Okay, so now another contributing factor is this hydrophobic box that is created by the presence of the tyrosine 188, tyrosine 195, and the histidine, as it helps to stabilize the dilysine trigger. Okay, so that's another contributing factor that helps to improve this this sort of multi-component stabilization. So just a few reminders about hydrogen bonds. So they can come in the form of a linear hydrogen bond, where you essentially have this sort of linear arrangement between the donor and the acceptor. In biological systems, that's not necessarily going to be the case. You most likely will have sort of a bent affiliation between how the proton is being shared between the donor and the acceptor. All right, so typically when we're referring to this arrangement, the linear arrangement is considered to be the stronger arrangement for the hydrogen bond network. But just to give us a perspective of distances as they correlate with bond strength. So a typical donor-acceptor distance when we're talking about hydrogen bond interactions is of the following. When you have from 2.2 to 2.5 angstroms, here we're talking about a relative strength that's almost at the level of a covalent bond, even though it's not necessarily a covalent bond. And then the corresponding bond strength, 40 to 14 kilocalories per mole. As you go further away, that becomes less covalent in nature, more electrostatic. And then you go even further away, it becomes quite weak. But often hydrogen bonds, when you have an extensive network of them, really helps to stabilize things, even if individually they may not be necessarily strong. Okay. All right, so this idea that the dilysine trigger helps to keep the metal bound closed in the end lobe, and then there are factors that are going to release and trigger the opening of the lobe. So that's why it's called the dilysine trigger. So at one point, it was assumed that if you were to protonate the lysines, that's going to open them up. And so that in theory is correct. But it's not as straightforward as just dropping the pH and it's going to open. There's going to be different factors that's going to contribute to this. But this idea of triggering the opening by decreasing the pH and protonating the lysines. It turns out that iron can actually be bound in the iron binding site in a partially open conformation. And what do I mean by that? Well, previously it had been thought that you're either iron bound, closed, and not iron bound and open. But crystallographic evidence has indicated that that's not necessarily the case. So this particular crystal structure that came out in 2012 is seen as sort of a snapshot, a potential snapshot of how you might have this sort of transient state of the iron being in an open conformation. Obviously, a crystal structure is not transient, but it captures a moment in time. This idea of something that's going to keep the iron in a partial open. And so if we look at this structure, you see that the iron has this anion, the sulfonate, that's coordinated to the iron in addition to the carbonate, the tyrosines. So in this particular context, it looks like this sulfonate is helping to satisfy the coordination while also maintaining the iron open because it to an extent, prevents coordination of the aspartate and the histidine, right? That's something that's significant here. So this idea of just pumping in protons is not necessarily the key, the sole thing as to releasing that metal. There's going to be other factors involved. And it could be something like chelation playing a role in terms of removing the metal. All right, so let's look at the C lobe, and we're going to see something similar, not identical, but similar. And so we have what is referred to as a pH-sensitive triad. All right, so where is this located? So it consists of these lysine 534, this arginine 632, and this is aspartate 634. And so essentially they are interacting with one another via an electrostatic attraction. And so you see where they're located with respect to the actual iron binding site in the C lobe. And so what I have on the right here are crystal structures for that triad, so the 3 different amino acids, as you can find them in the human protein, in the pig protein, and the rabbit protein. All right, so there are differences. So you can see in the pig protein and the rabbit protein, you have this really extensive interaction between the positively charged residues with the negatively charged atoms. And you see some amount of that, although not as extensive in the human one. But the clear thing is that electrostatic attraction between those 3 amino acids is key to helping to maintain this site stable. Okay, so again, similar but not identical to what we saw with the dilysine trigger in the N lobe. And you also have, what we mentioned previously, the hydrophobic box that's facilitated by the tyrosine histidine that helps to stabilize this electrostatic attraction. All right, so let's look at this a little bit more in depth, this idea of iron binding and how it can stabilize this protein, returning to this theme here. And so normally when we think about proteins, especially as you were taught in biochemistry, you normally think about their active form or their folded form and the denatured form. So you normally think about them in 2 different ways. But it turns out that it's more complex than that. And to an extent, you would have explored that if you look close at the hemoglobin model. Well, here, we're looking at this very simplified perspective. You have a folded protein, which would be the most stable form of the protein, and then when it undergoes some sort of transition. So this transition can be something that is chemically induced. It can be something that's thermally induced. Not many proteins can survive an increase in temperature. All right, so then that can lead to a denaturation event. All right, so this is a simple model of how you go from folded to denatured. Well, let's look at what happens in the iron case. So I already mentioned to you that iron binding in the traditional format results in a conformational change. And so what we're going to see is what happens to that protein. And we're going to look at this from a perspective of how temperature can give us an indicator of stability. So we're looking at a technique called differential scanning calorimetry. So that is a thermal-based technique where you essentially are ramping the temperature. So you can go from low temperature to high temperature. That's normally what you would do for proteins. Probably you're familiar with this technique, which is abbreviated DSC. but not necessarily for proteins, maybe for materials. All right. So they're comparable, right, in terms of how you're using them to gauge some amount of stability information. So the type of instrument you will use here would be different if you're looking for materials. So what are we probing here? We're probing the heat capacity of your system and how that's changing as you're increasing the temperature. So we're focusing here in this particular data is actually taken from this 1994 paper where the protein apotransferrin, so no metal bound, is undergoing this increase in temperature. And what do we observe? Well, we observe that there is an increase, this transition that occurs between, let's say, 55 degrees and 60 degrees. And then there's another increase in this heat capacity that occurs between 65 and 70 degrees. All right, so what does that do to? So when the authors of this work, when they assessed this actual work, they contributed to the unfolding process. The first transition, they attributed to the unfolding of the C lobe, and then they attributed the unfolding of the second transition to the end lobe. All right, so unfolding doesn't necessarily mean that it completely denatures, but it's undergoing a change in terms of its stability. All right, so let's look at what is referred to as the melting temperature, so the TM 57.6 and 68.4. And so that also tells us there's a difference in terms of even the stability of those two lobes. One is more stable than the other. All right, so now let's look at what happens when you bind iron. So we're doing the same exact experiment, but now looking at what happens with iron. When we add half an equivalent of iron, 1 equivalent of iron, 1.5 equivalent of iron, and 2 equivalents of iron. Remember, we need 2 equivalents or more to saturate the protein. All right, so what do we see? All right, so the apo, again, we got the 2 transitions right here. Okay, but then we add half an equivalent. So half an equivalent is not going to be enough to saturate even one site. But what do we see is that we get the feature here is decreasing, so that intensity is decreasing, and we also get a growth of a peak here. And the growth of that peak is at about, let's say, 85 degrees from what I can see here. Okay, so something is happening to the C site, and we see the growth of a new peak that's even higher TM. But then something really interesting is happening to the end site, and we see a transition where we're going from the original temperature to a slightly higher temperature. All right, so what happens when we add in 1 equivalent of metal? All right, so now let me just clean this up so we can track this. Now we're here. So what happens? Well, now we see that the C site is completely no longer there. Well, what does that do to? Well, presumably it's due to the iron saturating the C site. And then what else? Well, now we have the full formation of this peak over here. And that's presumably because the iron is bound to the C site. And another thing that we can see, the end site, is that this transition here is higher than it was initially. So what could be the reason for this? Well, one possible reason for this is what is referred to as cooperativity. That's something that when you were discussing the hemoglobin model, you discussed cooperativity, this idea that once you start binding oxygen to each of the monomers of that tetramer, it improves the capacity of the oxygen binding to the subsequent monomers. So there was a cooperativity, a communication between the monomers. Here, there appears to be a communication between the lobes for iron binding. All right, so then you add another 0.5 equivalent. What happens? Now we see that the end site transition decreases. Now this becomes more profound, okay? And then finally, when we fully saturate the protein, now we only have this one big transition right here, okay? Although that looks like one transition, the reality is that it's not one transition. It's actually a composition of transitions, right? But collectively, because it's pretty broad, right? You're going from a pretty wide temperature range. I would say you're starting from here all the way to here, right? That's about 15 degrees. There's a number of things that are going on, but collectively, that protein, when it becomes iron bound, is much more stable. So just a recap of everything that I mentioned so far, So we have an increase in the melting temperature of the C lobe of virtually 30 degrees. That's very profound. Even an increase of 5 degrees is considered a big increase. So you can imagine a 30-degree increase. And then for the end lobe, you have an increase of about 90 degrees Celsius for the end lobe. So it's quite significant what the iron binding does to the protein. Okay, any questions? Oh, go ahead, Javier.
I have a question.
Yeah.
Why does it start binding to the C instead of the.
Okay, so there's a little bit of a manipulation here. So it actually can be compound specific. So in this experiment, they use a form of iron, that's iron NTA. Okay, so this form of the iron, for whatever reason, the NTA allows for selectivity so that the iron binds first to the C site and binds then to the N site. My understanding is that the primary binding site is actually the N site for the metal, but because of the ligand, there's some amount of selectivity. If you were to change this to, let's say, iron citrate, this experiment would be much more complex. The iron citrate compound, I believe this is the iron citrate compound, it actually has no selectivity. So if you were to put the first equivalent of iron, you would see that both of these would actually decrease. Okay, and so then you will see the growth of the peak over here. Okay, so for whatever reason, the actual ligand facilitates some sort of selectivity, and that's something that hasn't necessarily been fully explored.
I think there's some iron in product that we see.
That for sure can be a case. Like there's a higher affinity, but remember, if you look at it for primary coordination sphere, it's identical. It has to do more with what's going on in secondary sphere and possibly something that the ligands are facilitating. Because it's known that citrate, at least citrate, can actually bind all over the protein. So they can maybe facilitating some interaction that prevents selectivity, whereas maybe NTA doesn't do that. Okay. All right. So the take-home message of all this. So this idea of how you alter the structure of the protein can also increase the stability. So not necessarily the native form of the protein is its most stable form. As we see in this case of the iron binding, actually affords more stability to the protein. And so here is data from the very same paper that we've been looking at, where you look at what happens to the protein in its end lobe in terms of its conformation. Here you have the apo fully open. We have the iron bound in the open form, but not as open as the apo. And then we have the more classical form of the iron completely closed. And it corresponds to an increase in stability. Okay, so this stability can be gauged in a few different ways. The example that we've talked about so far has been in terms of the thermal stability. All right, so here I want to emphasize this terminology that you can come across when it comes to this protein. So what's the typical binding? What I mean by that is iron bound to the 4 amino acids, the tyrosine, the aspartate, the histidine, and also the carbonate. That's referred to as the canonical binding. So initially it was thought all metals bind to this protein like the iron binds. But more and more, especially evidence from crystallography shows that metals can also bind in a non-canonical fashion, just like iron can bind in a non-canonical fashion, depending on what else is available. So here's the example of the sulfate group and how that enables iron to bind in a semi-open conformation. All right, so what is the net effect of this interaction? So we already talked about iron stabilizing the protein stabilizing the iron, and there's a corresponding increase in concentration, although I've already mentioned this already, where if you were to look at the concentration of iron in our blood and also in our cells, we have about 22 micromolar in blood plasma, and we have up to 10 micromolar, although that number can go a little higher within the cellular environment. All right, so let's look at other details of the transport process. So here again, we already discussed this idea of dietary intake. We talked about then the transferring in terms of it actually binding the metal, just binding the metal. But we haven't talked about transport, so that's where we're going to go next. There's some numbers that I want to draw your attention to. So dietary intake, let's say 10 to 20 milligrams you intake, of which you absorb 1 to 2 milligrams, okay? And then at the end, you lose 1 to 2 milligrams. So, and the net is that you're going to have a balance of iron. You're not going to have a surplus, you're going to have a balance. But in this process, you're going to have iron that once it gets bound to transference and gets biodistributed, it gets biodistributed in a few different ways. A large portion of that will bind to the hemoglobin. That's how we're able to breathe. A good portion of this will get bound to ferritin. That's how we store iron. And a good portion of this is going to get utilized and what we use for enzymatic processes. And another really, really important thing to note is that there's actually no physiological excretion of iron. The only method that all humans have for removing iron is by shedding. What I mean by your skin actually being lost, okay? So by shedding, and then you also have the menstrual cycle, which is another form of shedding iron. But typically there's no actual molecular mechanism, which is one of the reasons why it's important to control your intake of iron.
So.
Yeah. Go ahead.
So.
Yeah. Yeah.
So.
So so I'm going to talk a little bit about like how your body has some mechanisms to control slight excess and slight decrease, right? But like any excess, then you're gonna have to start thinking about chelation therapy, right? And then decrease, that's where you start talking about supplement, either dietary or if for some reason you're not absorbing properly, maybe some sort of parenteral nutrition, things like that. All right, so the transferrin bound iron is a dominant iron form in the fluid part of blood. And then there is some amount of non-transferrin bound iron, which is abbreviated NTBI, that can exist, but it's something that's very well regulated. All right, so let's talk about this process of the transport by transferrin. And so here I have a list of what I call the key players in this process. The apoprotein, right, which has to grab the iron-bound form, but also really importantly, the receptor, the transferrin receptor. There are two main forms, the receptor 1 and receptor 2, typically refer to the receptor 1, and how that receptor will recognize the iron-bound protein and how it facilitates the process of endocytosis. And so we have these additional key players that participate in the endosome, how they enable the movement of the iron out of the endosome and into the cytoplasm. So we're going to see a visual in a moment. But I'm going to highlight a few key events, and we're going to keep them in mind. So it turns out that the affinity of the protein, metal-free, iron-bound, is pH dependent. Okay, so at pH 7.4, apo protein, very low affinity for the receptor, but the iron protein, very high affinity for the receptor. Then when we're talking about the endosome formation, this endosome is actually a clathrin dependent process. So do you remember when I referred to clathrin? Oscar, do you remember when I talked about clathrin in our previous section? Clathrin. Does anyone remember this? So that protein, okay, that protein is a marker. Is a marker for the endosome to signal this endosome needs to be kept protected, okay? So that means that the endosome, when it brings in the transferrin, is not going to get proteolyzed. Proteolyzed being cleaved by proteins, because the cells want to recycle the transferrin to produce more cycles of iron being brought in and then the receptor being brought back to the membrane. Another key important is the actual endosome process, where there's a critical pH change from 7.4 to 5.5. That's what I was saying to you, that the pH of 5.5 is actually really important. And this is what we're going to focus on here. And then the steps of release of the metal from the protein, and then the iron escaping from the endosome, but escaping as iron 2+, no longer as iron 3+. All right, so these are the steps we're going to keep in mind. All right, so this idea of the pH dependence. Well, it turns out that in the membrane is where you can find the cell membrane, the transferrin receptor. So this receptor here, as depicted, you have this exodomain that can recognize the iron-bound form. It has a high affinity for it, and it can actually bind two molecules of the iron-bound transferrin. And you can see that it has this transmembrane domain. So I'm drawing your attention to the pH dependence. So in terms of its binding to the transferrin receptor at a pH of 7.4, the iron transferrin has a Kd value of nanomolar affinity. So Kd can sometimes be a little bit hard to keep in mind, because Kd is the opposite of Ka. It's the inverse, okay? And so you want this number to be lower and lower. The lower the number is, the higher the affinity is. Well, it turns out that at pH 5.5, it is the apoprotein and not the iron protein that has a very high affinity to the receptor. So now the apoprotein has a nanomolar Kd for the receptor. Put this as Kd. Right. All right, so let's look at two proposed mechanisms, because this is something that people still have some debates about, this process of how does transferrin receptor mediate the endocytosis. So the key players all remain important. It's not as if key players switch out depending on which method. But here we're going to look at how a chelator can play a really important role. So what we're looking at here is this idea of the pH decrease and how a pH decrease is not enough to release the metal. Because that's something I've already emphasized previously. So there has to be something that couples with the pH to help release that metal. And that's where a chelator plays a role. So it turns out something like maybe citrate or ATP. ATP is a really important molecule in our cells. And we've talked about it already in terms of how it facilitates energy. Well, it can serve as a chelator for iron. So maybe pronation coupled with chelation can help the iron to be released from the protein. And then once that iron is actually released, so dissociated, there's a protein or an enzyme in this case that can reduce the iron from iron 3 to iron 2. And the protein that has been identified is this protein called STEAP3. It's a ferrite reductase because it reduces the iron from iron 3 to iron 2. And then again, as I mentioned to you previously, the divalent metal transporter. So we saw how that played a role in terms of bringing iron 2 into the intestinal cells. It also plays a role here to release the iron from the endosome and into the cytosol. And it releases the iron as iron 2+, because as the name implies, divalent metal transporter, and iron 2+ is divalent. All right, so now let's look at some pictures to help us to make sense of what is it that we're focusing on. So here's a depiction of apoprotein and iron-bound protein. And so it turns out that two equivalents of the iron-bound protein can bind to the receptor. When it binds the receptor, it triggers that endocytosis process. So that endocytosis process results in this, the formation of the clathrin covering the endosome, okay? So that gets brought into the cellular environment. And then what we see here is this proton pump that's bringing protons into the endosome. And what we see is a change in the pH. So we were originally at a pH of 7.4, but now we are at a pH of 5.6, 5.5, not a big difference, okay? So 5.6. So then that pH decrease plus the presence of some sort of chelator results in the iron being released as iron 3+. All right, so we have the iron 3+, and then there's a critical reduction event where the iron 3+ gets reduced to iron 2+. Okay, so now the iron 2+ has been primed, and now it can actually transport through the divalent metal transporter. Here's the divalent metal transporter as depicted in that endosome. So that is the process of the chelator-driven process. Chelator coupled with acidification. So then you get that iron released into the cellular environment, and it transiently forms what is referred to as a labile iron pool. All right, what does labile iron pool mean? What do you think? Exactly, it's very reactive. So that labile iron pool always exists in our cells, but it's constantly being replenished. So iron comes in, participates in that pool, some of that iron goes to get stored, some of that iron gets used. But there's always going to be some amount of labile pool. And so I would say more and more is appreciated that it's not just iron 2, it's also iron 3, but there's more iron 2 present in the labile iron pool. All right, so let's look at another possibility of how the iron is released. So this possibility is by a lowered metal affinity driven. So what's driving the lower metal affinity? So here we're going to focus a little bit on the reduction potential, the capacity of the iron to get reduced to iron 2. So we already talked about how iron 2 has to happen, it has to form for it to get released from the endosome. But the question is, when does it get reduced to iron 2? So here I'm providing you this, this picture of what is referred to as the biological redox range. It's pretty wide. Some of this is not applicable to our cellular environment, but you can see biologically it's pretty wide. And we get a sense of things that might be able to serve as an oxidizing agent, things that might be able to serve as a reducing agent. So I mentioned that because we have to think about what's available in the cellular environment to reduce iron.
So when you worked, and this has been done experimentally, take the iron bound to the protein and measure its reduction potential. And so if we measure this value versus the NHE, that's the normal hydrogen electrode, that's the standard that you compare it to, you get a value that's negative 0.56 voltage. So that number, in terms of our perspective, is actually very negative. Okay, so what does it mean very negative? Well, a number that's very negative means that that oxidation state will remain basically iron 3. So it'll be very, very, very difficult for you to form iron 2 without reduction potential. But other people have studied, well, what about iron transferrin bound to the receptor? Is there a change? And it turns out, yes, there actually is a change in the reduction potential. So this complex here, what we observe is a reduction potential that now is negative 0.3 voltage. So it's more positive. Now we're in a ballpark region that actually can facilitate the reduction of that iron from iron 3 to 2. There are actually reducing agents that can facilitate this process. So this was rationalized as a possibility that maybe when the transferrin is coupled to the receptor at the pH of 5.5, maybe that's enough for the iron to get reduced to iron 2, and maybe that reduction event is enough for the iron to be released from the protein. All right, so possibly the STEAP3 can somehow facilitate the reduction of the iron, because it's still, STEAP3 is what's believed to be the enzyme, even when it's directly bound to the protein, to the transferrin, which is bound to the receptor. So why is this even significant? Why is it something that people have even thought about? Well, it's because the affinity of iron 3 to transferrin and iron 2 to transferrin is super different. You can see how different in terms of the log beta value, where iron 3 bound to transferrin is a value of 43.5, iron 2 bound to transferrin is a value of 13. And so it's believed that if the iron can get directly reduced when bound to the protein of the receptor, that reduction event will make the iron get dissociated, and now the iron 2 can freely be transported by the divalent metal transporter 1. So people challenge this model. They challenge this model because the question is, how exactly can STEAP3, which is a membrane protein, come into enough contact with the iron center to facilitate the decrease in the reduction of the iron from iron 3 to 2. So that's one of the things not in favor of this particular model, but something that seems interesting in terms of the possibility. So just to review this model, so basically we have this idea of the iron-bound transferrin. You have that affinity constant, and then you have the reduction of the iron, how that gets fine-tuned by the presence of the receptor from negative 526 to negative 300. Then you have the iron capable of getting reduced to iron 2 plus, and once reduced to iron 2 plus, it can on its own be liberated from the protein and then transport out of the divalent metal transporter, right? So of these two models, I would say that the first model that I gave you is considered the more likely model. All right, so the question people are exploring is, well, what about non-ferric metal ions? Because, as I already mentioned to you, transferrin has the capacity of binding other metals because it is a hard base. So it can bind hard metals. And there's evidence to support that it does have the capacity of binding other metals and maybe do so as a natural process of its functions. But that's something that's still debated, whether it is natural for transferrin to transport other metals, or is that just a product of introducing other metals into our body? All right, so here are three proposed modes for non-ferrics, that means non-iron metal, serum transferrin entering into cells. You have possible binding either canonically, like iron will bind in this typical format, or non-canonically at the metal binding site. You can have binding in a mixed metallation. This is something that a few people have proposed in terms of what has been proposed for, let's say, plutonium, which is actually considered, which is a toxic metal, but possibly this idea that you might have a mixed metallation where you have iron in one lobe and another metal in another lobe. Or a third possibility is considered advantageous binding. What do I mean by that? Well, let's imagine we have iron bound to the protein, So then you have a conformation of the protein to which another metal or metal compound will bind. And through that interaction, that metal or metal compound can enter into the cell. So just basically taking advantage is riding on the iron-bound transfer in order to be able to enter into the cell. All right, so this is where we're gonna end off with this note. So there could be at least two molecular mechanisms for endosomal release of non-redox active, and I'm gonna just emphasize that, non-redox active hard Lewis acid metals. It could be that there's acidification with chelation that leads to transport via the divalent metal transporter. But that would assume that the metal will have to be divalent. That's one model. There's also another model where you have acidification, chelation, and then that chelator serves as an ionophore. Remember, ionophore means small molecule that transports. So if the chelator itself binds the metal and it transports it out of the endosome, right? So that is a possibility. And that's summarized in this picture right here. All right, so any questions before we head out? Any doubts? So esta parte, la última parte is still under study. It's proposals. Pero el del hierro, that's something that is well characterized. Okay. Any other questions? All right, everyone. Go ahead. Do you have a question or you just want to see it?
So it's like.
You can look at it. I'm gonna just stop. All right, everyone. Take care.